that primary cancer is actually viable. It contains dividing cells. It's not full of necrotic debris. And so if you look under a microscope to compare the two, the tumor under the skin, very little stroma, lots of blood vessels that are right next to the cancer cells. You look in the primary mouse tumor, lots of stroma, blood vessels far from the cancer cells. In the human, it's even more pronounced, lots of stroma, blood vessels far away. We quantitated the number of blood vessels, and it was about 25% of what you would have in normal tissue. And so this flies in the face of the concept that angiogenesis is a requirement to promote cancer. And we didn't believe our data when this occurred, and so we employed other individuals to help look at this. And so one bit of that data is shown here. Collaborators from Johns Hopkins, Chris Iacobozio's lab, showed that inside the pancreas tumor, the mean vessel density was 10% that of around the tumor. So it had dropped a factor of 10. And this was in stark contrast to other types of cancer, such as colon cancer, where there was about a log difference in mean vessel density. Pancreas tumors are white and hard. The surgeons have known this for ages. For some reason, the oncologists and the pathologists have never really looked at the tumor critically before, and so we were obviously stunned by these results. The vessel distance between the vessels in the cancer cells is 30 to 40 microns in the human, a bit less in the mice. But this provided us with the first glimpse into maybe why drugs weren't working so well in our mouse model, but more importantly in patients. We had this model that looks like the human cancer, and then it's not drug responsive, like the transplant models are. Two cytotoxics we looked at, gemcitabine and adria, were poorly delivered in our model system. And we made the surprising observation that pancreas cancer is hypovascular and correlating with stromal abundance. And so the concept came that maybe we could do something about this if we could get rid of the stroma. Maybe the drugs could get in. And fortuitously, the other therapeutic project which Ken was engaged in was looking at a hedgehog inhibitor. And this was one that was provided to us by some colleagues which are only located a mile from here, Infinity Pharmaceuticals, which is a chemical derivative of cyclopamine, which is a very good inhibitor of smoothened. When Ken used this drug, he had some very dramatic results that suggested to us that this pathway may have a role in the paracrine or stromal compartment. As he was doing these experiments, Fred de Sauvage published, I think, the seminal work on this, where he showed that if you use a reporter mouse where the pancreas cancer cells are green and the reporter cells on the outside are red because they have a patched LAC-Z reporter construct. Patched is a downstream target of GLI, which is a transcription factor in the hedgehog pathway. These cells are always on the outside. They're never found within the neoplastic cell compartment, suggesting that the actual active signaling was on the outside. Doug Hanahan corroborated that by knocking out smoothen and turning on RAS in our model. What Ken was seeing at the same time was that when he was treating with the hedgehog inhibitor, the stroma went away. So here's treating with salt water, here's gemcitabine, hedgehog inhibitor, the stroma is gone within 10 days. These are all cancer cells. And if you include chemotherapy when you do this, the cancer cells are dying. So these are fragmented nuclei. In parallel, if one looks at the density of blood vessels just by using immunohistochemical detection of CD31, you note that these very few blood vessels become more numerous and more open. And so when you look at the doxorubicin surrogate for drug delivery, you note that you're starting to get that green dye on. And so this suggested that maybe this would be an approach forward. And so Ken, with this information, was able to show then that we were getting more gemcitabine into those tumors, treating for 10 days, more cancer cells were dying, and most relevantly, more mice were living. And so the purple curve is where you're combining hedgehog inhibitor with gemcitabine. These other colored curves are all the monotherapy and controlled combinations. And so this was interesting. You shift the whole curve to the right, you probably had a general effect. The mice in the end do die, but when he looked at the mice, he actually had a huge impact on metastatic burden. That was decreased dramatically. And he tried to understand resistance, and he couldn't. If he tried to study, again, gemcitabine transport or gemcitabine detoxification pathways or the targets of gemcitabine. But when he looked again at the blood vessels which had come up in number now receded. And so there was an adaptive response by the tumor and the tissue somehow to this sustained treatment. which was a potential mechanism of why it relapsed or became refractory in the end. And so what Ken could conclude from this early work was that poor drug delivery really is a primary problem in pancreas cancer in a mouse, and potentially also in our patients, and that methods that improve tumor perfusion could circumvent this. But one question is the hedgehog inhibitor the only way to do this? And what will this matter in terms of how can we translate such findings into the clinic? So the first bit of work, Ken had a tremendous number of collaborators from around the world, some in Dresden, some in various parts of the States, the pathologists at Hopkins, our colleagues at Infinity, as well as lots of funding agencies, and the majority of the lab helped out a lot on this. And so just to answer some of those simple questions that I added on that last slide, just in snap order, Mike Jacobitz, who's a grad student in the lab who worked very closely with Ken to get the animal therapeutics effort off the ground, was able to show with an extracellular matrix enzyme that he could infuse intravenously, which is well tolerated by animals. And again, I'm not sure that maybe the folks, if they can turn on the lights, that would be helpful. This is a blue spot, and here's the green tissue. Here's a pseudo-volcano plot of that. If you infuse this extracellular matrix enzyme in the bloodstream, very rapidly you lose your edge, the barrier, and actually you can get drug in an hour later. And so Mike was able to use this approach in a different way than the hedgehog inhibitor to actually open the vessels and get drugs in. And so he could show an hour after infusing the drug that the vessels, which are not just few in number but also compressed, you could pop open more than 2/3 of them. So it was 33% were compressed, 66% were now open when you infuse this extracellular matrix digesting enzyme in. And with this approach, he could actually get a therapeutic response that mirrored and maybe was slightly superior to that of the hedgehog inhibitor, shown here by tumor velocity plots, and here by a survival curve, where here's the combination treatment. Again, the mice do succumb at the end, and we're trying to understand that now. But I think that provides another sort of nail in the coffin that drug delivery itself is probably going to be a primary problem in this disease. And there are multiple other ways that people can attack the stroma in this disease and others that may be germane in the treatment of these patients, as well as those who have fibrous-rich other types of carcinomas. In terms of translating this into the clinic, though, we first need an actual practical way of doing so. And so the other surprise I had by going down to the pathology lab is shown here. And so I went down to see what the biopsies would look like if we were to enroll patients on a short-term clinical trial. And these were the slides that I was given. And I went back and I said, there's a mistake. These are from a pap smear. And they said, well, no, this is actually what they look like. Because cytology work, as some of you probably know, is really based around the eminent work of Georges Papanicolaou, who is a Greek-American pathologist and physician. This approach is used for tumor aspirates, which is the classic way by which pancreas cancer is diagnosed by endoscopic ultrasound. So we looked at this, and we knew we couldn't really learn anything scientifically from such things. And so working with the pathologists and the surgeons and the gastroenterologists at Cambridge, we actually, with the same size needle in the EUS, we were able to get something we call microcores, fix them properly, stain them with H&E, and this is how big they are. And here's your cancer cells surrounded by stromal cells. We can actually do laser capture on those stromal cells and measure things like GLI1, et cetera, and do the trials that you can imagine based on this preclinical work. And from a medical economics as well as scientific standpoint, this is a far superior approach. And so we're hoping that this is going to be useful not just for our crowd in England, but potentially elsewhere. Okay, so one point about pancreas cancer learned from a mouse is that drug delivery may be limiting. So gemcitabine gets there in suboptimal concentrations. We need to raise the level of gemcitabine. But as you've heard, throughout the last day and a half, a lot of excitement surrounds this concept of oncogene addiction and what are the pathways that we can go after based upon the mutations that the tumors have. And so we had a variety of other approaches to look at this problem, and I'll show you snapshots of each. In the first one, Pedro Perez Mancera has been working on doing forward screens in our system by looking for genes that cooperate with oncogenic KRAS to promote pancreas cancer, with the hypothesis being that these are going to be pathways that potentially will represent vulnerabilities that could be exploited in the developing or established tumor. And so he used a system which was designed and implemented by Dave Large-Espada, introduced in this forum, I think, a few years ago by Nancy Jenkins. But he also built his own conditional mouse. And so in this mouse, the transposons are hopping in the whole mouse. And in this mouse, they hop only in the pancreas, where you have Cre expressed and you're turning on the RAS gene expression. And again, the transposon technology is something that most of you are familiar with, but if you're not, that's how Indian corn exists. The jumping genes were something that Barbara McClintock described years ago. They occur in many species, fortunately not in humans, and they've been hijacked from either cabbage moths or, in this case, trout, to use as a genetic tool. Pedro was able to show with the sensitized transposon mice, the ones that are hopping constitutively in the soma, that he could get cooperation in pre-neoplasia and pancreas cancer very easily. So here's the comparison of just KRAS in the pancreas. Here's with the transposons at 3 to 5 months. A few months later, more, and at the end stage, more. And so again, the bars are showing you more advanced pannin accumulation. In a similar fashion, in the conditional mouse, he was able to show cooperation for lethality. And so the reason why you do these experiments is you can go back and identify the site of integration of the transposon, which is a technique that I think was really essential prior to the advent of the more sophisticated and exciting genetic sequencing approaches you heard about from Eric Lander. So what do you expect when you do this experiment? Well, you expect to get what the human database tells you about pancreas cancer, these tumor suppressor genes that cooperate with RAS, because again, this was a screen done just with the RAS background. But that's not what we got. Instead, we got a list of genes which, in general, are genes that activate the RAS pathway more, that turn on the pathway more, that turn it up. The first gene is a dub, which I gave a funny name, PFG1, for Pedro's favorite gene 1. It's not because I'm afraid at floor meetings to tell you guys about it. It's just that we still don't really understand exactly how it works. I mean, it clearly activates receptor tyrosine kinases in both pathways of the RAS pathway, but I suspect it does other things. So it's found in 57 out of 160 samples. 30 to 40% of the samples have it. It's never been mutated in human cancer that we can tell, but it's been fingered by many to be important in cancer initiation and progression. The other main hit we have is in the PI3 kinase pathway, PTEN, GSK3 beta, as well as MAGI1, found very frequently. CTNNA1 also. Mutated genes in human pancreas cancer are shown by asterisks. So ARID1A, which is a chromatin complex gene, as well as SETD2, which is a histone methyltransferase, are also found. But the interesting thing to me was we didn't find the tumor suppressor genes that we thought we would. And when you look at a cartoon of the PI3 kinase pathway, looking at PTEN, looking at alpha-catenin, which was the gene I was stuttering over, and looking at MAGI1, they all participate in this same pathway in activating the PI3 kinase pathway in addition to other things like Wnt signaling, et cetera. And AKT2 is known to be amplified in human pancreas cancer. And so you walk back from this and think, well, so what? You already knew that the PI3 kinase pathway was important in RAS signaling. You heard from Julian Downward yesterday that it was one of the two main pathways in RAS oncogenesis. But it suggests that maybe the PI3 kinase pathway is not active enough to help promote pancreas cancer. Now, Pedro was able to do so far some brief experiments in Matrigel with Si or more prolonged experiments in Augur with SH, looking at these various gene products. They all appear to cooperate in terms of promoting growth and or survival of cells that have mutant RAS. And so that was an interesting, I think, interlude to things that we may be thinking about in terms of designing therapeutic strategies.